moment.
Okay, no problem.
Okay.
Okay, we are on live now. I will pass Fabrício. More. Put in the room. Joanna, I will put you in the waiting room. Okay. Everything okay, Ada?
Okay.
Yes, I will you do a short introduction and you can start, right?
Can I can start?
I will introduce you. Let me see. Good morning, everyone. Welcome to the Darwin Day. And it's a great pleasure for me to coordinate the first Darwin Day conference. And I'm very happy to present to you one of the most important research in the area of origin of life. And her studies have completely changed our view on the basic function of the cell. And I'm sure it will be a wonderful conference. Ladies and gentlemen, Ada Yonath. Ada, thank you very much.
Wait, something happened. Okay. Shall I start?
Yes.
So, good morning, and thanks for getting up so early, just for me. I want to talk to you about origin of life, and I think that the ribosome is the connection between it and our life. And actually, I have another title, which is, oh God, which is ribosome and connection between the far past and the near future. So what is ribosome? When DNA instructions for proteins have to be transcribed, first of all, they are transcribed to messenger RNA, now it's very popular for the vaccination. And then this is translated by ribosomes to proteins. So there is a huge number of ribosomes that function in each living cell. Mammalian cells may contain millions of ribosomes. Even bacterial cells contain up to 100,000 ribosomes in a single cell. All ribosomes perform their two major functions, the decoding and the formation of the peptide bond, in the same way. Doesn't matter where they come from, which animal or which bacteria, and they act very fast. They can even make 40 peptide bonds in 1 second in vivo. It means a protein is a few seconds, and they hardly make mistakes. The mistake rate is between 1 to 500,000 to 1 to 1,000,000. They're all made of two subunits, small and large, that are connected by the tRNA that brings the amino acid. So the decoding center is in the small one, and the peptide bond formation, it's called PTC. It's peptidyl transferase center, it's in the large one. And tRNA is the molecule that is associating with the small subunit for decoding and bringing the amino acid to the large subunit. I'll come back to it in a minute. But first, maybe we want to see a little video that we made directly when we saw the structures. So we worked 20 years on the structures, and immediately we understood how the ribosome functions. So what you see here is messenger RNA is reaching the small subunit, and this interaction between them and going into its path is monitored by initiation factor, this blue thing. Once this is okay, the first tRNA can be brought by another factor, initiation factor 2. I didn't tell you, but you're looking at the ribosome of bacteria. In human, it's done the same way, but it's more complicated and larger. Now the tRNA is associating with the messenger RNA from one side for the decoding, and the amino acid, this little blue thing, is on the other side. And the two subunits will associate these bridges between them, and the active ribosome has been made. So you see it now from the solvent side, the small subunit below, the top subunit, the large on top. And the amino acids are brought by tRNAs with elongation factors. Each tRNA is elongation. So when they come in, the ribosome helps in, out. If they come in, they are decoding. We took away the large subunit, so you can see that the decoding is here, and that the peptide bond is made on the other side. In a minute, you'll see it better. And the newly born protein goes into the tunnel. Actually, this is the only thing of the large subunit that you see now. Just the tunnel, the exit tunnel. And now you see the whole ribosome again. The protein starts to come out and fold here, this blue thing. And the process continues until there is a stop codon. And then instead of elongation factors, there is recycling factor and release factor that will come in when a stop codon is there and dissociate the ribosome to the two subunits. The protein, this blue thing here, can go and do what it needs, and the whole system is waiting for the next job. That's all. So that's how ribosome works. So what you saw in the movie, two subunits are actually made of a lot of RNA. This is the gray thing here, and many ribosomal proteins. Each of them is shown in a different color. Small subunit and large subunit. And just to remind you, the small is decoding and the large is making the peptide bond. And that's correct for all ribosomes. Even in eukaryotes, when they are larger, they do the job the same way. So essentially what we saw until now is the ribosome. Now we look at it from the sort of front side, small subunit, large subunit, decoding here, and the PTC where peptide bond is being made here. And these are the anti-codon sites of the tRNAs. These are the free tRNAs, and there are free sites. A, P, and E is exiting. A is amino acylated as P is peptidyl, and this happens here in the PTC, as you saw in the movie. So we looked at the PTC, and we saw that there is an almost identical semi-symmetrical region at the PTCs in all non-ribosomes, bacteria, archaea, eukaryotes, mitochondria, and you can see here where you can get the coordinates from them in the PDB. All of them have, if we look at the main chain, almost completely the same. Very small, very, very small changes. And it's semi-symmetrical around the center here. You can see better from the side. So from the side, what you see here, actually you saw it here too. The piece that associates with the A site tRNAs in blue and the P site tRNAs in green, and I will keep these numbers, so blue and green. And you can see here the end of the two tRNAs, and I'll come back to it, but they are sitting the same way in all ribosomes. Just to show you the level of symmetry, so we put the A site on top of the green site, round around an imaginary axis. It is shown here in red, and you see how much they are similar. They are not perfectly similar. Of course, they cannot because it's all L amino acid or L RNA. It means there is a chirality, but the main chain is almost the same, and you can see here pieces that even the side chains are superposed on each other, except for this one, which is an important one, 2602. I'll come back to it. So the whole site is sitting between essentially all the active sites of the ribosome. It is connected to the decoding by the tRNAs. Here you see only the large subunit. And the two hands, you remember in the movie, the two moving hands, it's connected directly by continuation of the RNA. So they are connecting between everything that is needed to make peptide bonds and transmit them. So the high conservation of the symmetrical region indicates that its existence is beyond environmental conditions. Therefore, we propose to call it the proto-ribosome. That it was there before anything, and it's still there. So essentially, no evolution on it. And this proto-ribosome is a prebiotic bonding entity, as before, that was termed by us, and it functions in the contemporary ribosomes too. So based on the suggested existence of an RNA-dominated world, and I think it's not my idea that there was an RNA world, and on the findings that RNA can replicate and elongate itself and has catalytic capabilities, we propose that the proto-ribosome is the entity around which life has evolved, even before life. And there are here several references to show that the RNA can function under prebiotic conditions. Neither of them is ours, and I will not now talk about each of them, but Joyce and Shostak and Sutherland, they all showed this in their work before us. So possible pathways of evolution are based on the existence of RNA world and on the finding that RNA can replicate and elongate itself. This is in our opinion the way it started. When there were already amino acids, the peptide bond formation, just to remind you, is made by the two tips of tRNAs, which are already connected to the amino acids. It means there is no water to go out. The water has gone out by the connections to the A site tRNA that brings a new amino acid, and the P site tRNA from which the peptidyl is growing. This is the reaction. That's all. Very simple. To remind you what is tRNA and what is important for us today, it's always this shape, this L shape RNA everywhere. It always has an anti-codon loop here of 3 nucleotides that will bind to the codon, and at the other end, CCA amino acid, which is fully conserved, fully conserved, CCA amino acid, and this is the part that binds to the PTC. Some changes in the tRNAs are here. Of course, each amino acid has at least one tRNA, sometimes they have several, but they always have this shape, this L shape, as you can see here. How do they sit in the PTC? So in the books, they like to show them in biochemical books that the two tRNAs are parallel to each other, and they read the genetic code, which is connected to the small ribosomal subunit. This is correct. And then from one of them, the protein, the newly born protein, comes out. This is really wrong, because how will this amino acid sit this one? They have to sit this way. They have to see each other and make the bond. And I have this fight with with all teachers, but this is the way it is, and this is the way it is. So you can see here the active site, the PTC, and the way that the tRNA sits. You see, in a way that they will see each other. The ribosome here is in gray, and A site in blue, and P site in green, as I keep all the time. And here, this is the PTC, the active site. But they always sit, so they meet in the active site in an angle. And this is dictated by the ribosome structure. So what I did in order to convince you, I took all types of substrates from, not only from our lab, from other labs, from Yale, for instance. And I superposed only the active site components, not the active site itself, just the A and the P site tRNA, just the substrates. One substrate was really very long, and here another substrate was long to the other side, but it doesn't matter. It's always sits like this, dictated by the PTC structure. So when we looked at it carefully, and when we took into account that there is no water to have to come out, actually there is no catalytic activity of the ribosome. It just provides a frame. All the catalytic activity is peptide bond formation. It's an exothermic reaction. It happens on its own once the geometry is correct. And the ribosome makes sure that the geometry is correct. You see here, the ribosome is in gray, and the tip of the A site and the P site tRNAs are shown inside. You see, they cannot do anything. They must sit here and make the peptide bond. Once they are there, they are there. So essentially, not only that the ribosome architecture makes the peptide bond formation, but also it hints on its origin. Actually, the whole region is 3 to 4% of the total ribosomal RNA. Depends if it's a prokaryotic or eukaryotic. But this is the same. You can see it here in blue and green, and it is kept, as I said, and it's functional also in the contemporary ribosome, and it hints that this is the way it was created. So once we looked at how it's built, the question to answer is, how is the peptide bond formed? So in books, you hear that the peptide bond is formed in the P site and then goes to the A site. It means if in the A site there is already an amino acid ready to go, and the protein is already so long, it goes all the way to the A site, then the A site is put in. Is it clever? Isn't it more clever that the A site goes to the P site? So there was a long discussion in books. I can still see that there is dispute, but what we did, we really looked at it and we identified the tRNA motions from A to P. So we think that there is a shift of the helical part and the rotation of the 3' end. And I will show you what we think. So when we looked at it, here is the A site. When we looked at the structure around the A and the P site, we saw that there is an empty space here. And only two nucleotides bulge in, 2602, which I mentioned earlier, I'll mention again, and 2585. So what happens in this empty space? We made a collaboration. We collaborated with the group of Massa from City University in New York, and they calculated what would be the path of the tRNA, of course, the end point of the tRNA from A to P. This is the empty space and the empty space. And they found, this was their calculation, that if this is the way, the transition state is almost at the P site. It can be created between the A and the P, just here. It means it goes from A to P and not from P to A. And how does it go? So if we look at it, this is the site of the A site, and this is the P site, and it goes like this. Chuk, chuk. How nice. You see, exactly. The ribosome is really made for it. A new one can happen. And that's really not only interesting, but it explained results that were observed in Japan, that these two nucleotides that nobody thought that they are important, they are very important, since mutations in them will disturb the rotation. So what is propelling the rotation? We think that I already mentioned the name 2602. So it's known that 2602 can have all these structures in different complexes. So we think that this is the propeller going from A to P. Very clever machinery. So 2602 may propel the rotation motion. So here again, just to remind you, small subunit, large subunit, here is the PTC, and this is the decoding. And if we looked at it as before, the components, the tRNA and the P site tRNA are sitting this way, or we can see it that way. The peptide bond will be made here, and the whole machine looks like that. Just to remind you again, so this is the symmetrical region. This is its size in the whole ribosome. This is its a shape from top, from the side, and this is what we think is the proto-ribosome. So we propose that the proto-ribosome is the entity around which life has evolved. I said it earlier. What are the possible pathways for this? They are based on the existence of the RNA weld and on the finding that RNA can replicate and elongate itself. What, where, how was it made? Was it in ponds, in the ocean, in the desert, in vesicles, outside? It doesn't matter. It can happen anywhere. But it doesn't disturb our assumption that it originated within an environment which could contain elements of life, and that the prebiotic chemistry could generate RNA chains having structures as in the contemporary molecules. So I don't want to talk much about this. You can read it if you want, but what's important is that it was shown that even structures like this of RNA can live within micelles. It means it doesn't really need a whole complicated weld to exist. And to remind you, this is the interface we talk about here, and tRNA will bind it to here. So we decided to construct the proto-ribosome. Now, after I talked so much about it, to construct it in the lab. And one of our hypotheses suggests that the catalytic proto-ribosome is a product of dimerization, and it requires the existence of self-replicating, self-folding, and self-dimerizing RNA molecules. So again, to remind you, we talk about here, or maybe not to remind, to tell you, this is the whole RNA map of the large subunit in the large part of the large subunit in bacteria called 23sRNA. And this here is the part that constructs the symmetrical unit, the proto-ribosome, or the active site. So here it is, and in order to build it, we thought that we should make components that resembled the P site, and the A site, and try first of all to see if they like to come together. And just to show you again, this is the part, this is the P region and the A region, and we try to imitate it in the lab in a test tube. So the earlier picture, you could see this part. Now I isolated it from the whole map. And we cut it like this. And we had this part and this part. On the assumption that there was dimerization, we made these two parts, but it could have been not just dimers. And I won't talk today about anything else, just dimerization. So our motifs, in order to construct it, could be like the stem, elbow stem, which is the main part of tRNA, but also the main part of the active site of the PTC. Because only the backbone and the orientation of the molecules of the RNA are symmetrical, and there is no sequence symmetry, we could play with the sequence according to what is found in different ribosomes. So the sequence may change, but not the position, not the symmetrical position. You can see here the conservation of the PTC sequence. Some reach 100%, some 75, 60, and some are 50. There is nothing that is smaller than 40 or so on, but most of them are highly conserved, but not fully conserved. So this is the way our way of thinking, from homo to heterodimers. So homodimers was relatively easy to make. It means taking only the P or only the A. But heterodimers were more difficult. And it seems that the proto-ribosome was a dimer of RNA chains with sequence resembling one side, and then they changed. But it only seems, and by now I'm not really sure that this is a very strong statement. So since RNA can be the template for itself, it's conceivable that the proto-ribosome was a dimeric enzyme product by either gene duplication or gene fusion. I'll talk about duplication. I'll tell you what I think, what are my problems with duplication in now. So gene duplication, why should it do it? The idea of duplication is that the molecule sort of knows what happens, but how one side does not know what happens with the other half of the substrate. Only half binds to it. So it's not really clear that it was a simple gene duplication. There is also a logical problem. Why should RNA construct a machine for polymerizing amino acids? This is an action that results in competitors that are better than itself. RNA enzymes are not so good. Protein enzymes are almost all enzymes. Why should it do it? So I'm not sure that RNA sat down and thought, but it seems, or what we think, that is that there was an RNA machine for performing RNA needs. This is the pre-proto-ribosome, prior to the appearance of the amino acids. And then when the amino acids came, they were so fast, they snatched it and turned it into their efficiency and beneficiency. So we are trying to see, first of all, if we can not only make dimers, but also see if they have binding capabilities. So just to remind you, this is the reaction between A and P site, and this is tRNA. And we are concentrating on its little part here, the CCA amino acid. So we use the fragment, what is called fragment reaction, that the biochemists like to use in order to see whether the ribosome is active. But essentially, it's two amino acids that make a D bond, peptide bond. And after 6, 7, 8 years, we finally got lab construct, not only one. Actually, we have now, I should add here S, lab constructs of the proto-ribosome with catalytic properties that they form peptide bonds. And if you want some detail, there are several like this. The smallest is 67 bases, the largest is 90 from each side. They were characterized by MALDI, and we had controls, positive control in presence of large subunit, negative, no reaction, and they were all fantastic. And the time is less than 1 second. I don't have the exact time yet. We are still improving the reaction. So what is the conclusion? That is a prebiotic chemical bonding entity is functioning within contemporary ribosomes in all living cells. So these studies gave us a tool to ask a question: what was first, the genetic code or its product? So actually to look at the emergence of the genetic code, and it's only a possible way. So the initial dipeptide, and according to our assumption, the initial dipeptide could be substrate for the formation of next peptide bond and so on and so on. And this way, oligopeptides could be formed, but not very long ones, those that could be held by the original proto-ribosome. But the existence of well-performing oligopeptides that could catalyze fundamental reactions or stabilize the machine could be the lead for the emergence of the genetic code. So what I mean, what are needed ones? For instance, this one that can carry metals like zinc, and it has some histidines that could bind to it. The question is whether histidine is ancient. I'll answer in a second. But what could be other substrates? Substrates could be charged tRNA and amino acids and so on. So what do I mean by stabilizing the machine itself? Making peptides that will sit in these positions and keep the machine together, because actually there is no bonds between these endpoints. Or what I showed earlier, and here I can tell you why I think that there was a histidine, although many people think that histidine is not one of the earlier ones. If you look at the abundance of histidine in all types of these enzymes, transferases, hydrolysis, and so on, you can see that histidine is the highest in appearance. This is given to me by Lascano, and maybe this shows that even in the prototype, histidine was important. It could be leftovers of partially destroyed RNA, so there is a reason for it to exist. So anyway, this idea suggests that the genetic code was created by or according to its product, which were fit and useful, therefore survived. And they led to the creation of the primitive original genetic code, which co-evolved together with each product. This is the conclusion from all what I said before, and I will repeat it. The genetic code co-evolved together with the ribosome as well as its products, the proteins. So if I can summarize, from proto to modern ribosomes, throughout the sequence of each component of the central region has been genetically optimized for its functional task. It means one is accepting and one is releasing. And it is indicating that function is superior to sequence preservation and explaining the lack of sequence symmetry in the symmetrical region. So ribosome has been and are still RNA machines. And RNA enzymes are known to be inefficient, lazy or lousy as you want. Nevertheless, in the contemporary world, the ribosome is an amazing chemical apparatus, one of the most efficient ones, or maybe the most efficient. This indicates that nature devised means to turn lousy machines into an efficient apparatus. And how does this happen? By addition of proteins to the ribosome. So therefore, it's not just an RNA machine, it's RNA protein machine. So you can see here the structures of all small subunit and large subunit ribosomal proteins. And you can see that all of them have long tails at their ends, or here you can see really beautifully, or internal loops. And in the structure, they go into the structure. You can see these four in here. They see in the loops are coming in. The globular parts are mainly on the outside, and they're keeping the RNA together and giving it the possibility to be amazingly efficient. So in a summary, our hypothesis is based on the existence of self-replicating RNA molecules with catalytic capabilities, and on the assumption that the genetic code followed its products. So I want to talk about another point very shortly, and I'll explain why they are connected. So owing to the vital role played by the ribosome, over 40% of the clinical useful antibiotics target protein biosynthesis, mostly by paralyzing the ribosome. That's very clever. You want to get rid of a pathogen, paralyze its ribosome. The natural antibiotics are the weapons that microorganisms from one type are using for interfering with cell life of different species. And most of the antibiotics, anyway, the natural antibiotics are made by bacteria themselves. And I want to show you how clever they are, the antibiotics, if it works. It works, yeah. So here you can see a small antibiotic called edein, found in Poland. It disturbs the connection of the messenger RNA. Doesn't let it bind. That's it. So small, so clever. Here you can see even a smaller one called tetracycline that occupies the position of the second tRNA. So one tRNA is bound, but the second cannot. So these two work on the small subunit. Now I'll show you one that works on the large subunit in the tunnel. It's the first one to be used after penicillin, called the erythromycin. It sits in the tunnel and doesn't let the protein go, block the tunnel. And the last one is called clindamycin, and in my opinion, is the cleverest. Just sits in the position of the bond. Have a look. How wonderful. How clever. Sure. That's it. So I showed you 4 antibiotics, but the problem I want to talk about is not how they work, but that there is resistance to antibiotics, and this is one of the most severe problems in modern medicine. I'm sure you all know, I don't have to explain it. How does it happen? This is something to do with evolution. During bacteria cell division, or by acquiring genes from other bacteria through horizontal gene transfer, there is a chance for mutations. which may be random, but also may result by a resistance, and it can even happen by radiation. So some mutations can lead to resistance of antibiotics for providing advantage, and therefore for population of resistance of bacteria that can increase by natural selection. And bacteria profit from accidental mutations to develop specific molecular pathways that cause resistance. It's really very interesting. But my question is, can we combat resistance fully? I don't think so. I think that bacteria will be always more clever than us. They want to live, but we can still do some things. And I want to show you one modest contribution and what can we learn about evolution from it. So our concept was to look at the pathogenic bacteria and the resistant ones and see how they differ from normal ones. So for this, we determined the structure of ribosome from pathogenic bacteria and compare them. And here you see a comparison. The skeleton of the non-pathogenic is in gray and the pathogen in cyan on top of it. Very small differences, less than 8%, but they all sit in specific places, specific points. Like for instance, this one, that for fighting resistance, it can be important, but I don't want to talk about it. I just want to show you that there are several like this in several places, and we looked at them, and this will explain you why did I connect the two topics. Why are antibiotics involved in evolution? We tried to see what are the additional parts that I just show you, the additional parts of the pathogens. We compare them to increase in size, in RNA size, of going from eubacteria to eukaryotes. The harmless eubacteria here are shown in orange, the eukaryotes in purple, and the pathogenic in cyan. And look, they're almost the same. The addition of the pathogens is like the addition with evolution from prokaryotes to eukaryotes, not in many places, but at least in some. So the question that I'm asking, as some of the extensions in the pathogen are similar as the extensions between prokaryotes to eukaryotes, the question is pathogenicity a missing link between the two kingdoms of life? Or in other words, are pathogens the missing link between eubacteria and higher organisms? I don't have an answer, maybe you do. Just to remind you, we talked about the proto-ribosome, and I want to thank the Weizmann Institute for letting me work many years on it, the Max Planck organization that created a group for us in order to determine the structure of the ribosome. It was initiated by Dr. Weizmann, that died much before we got the structure. I want to thank my two groups in Israel and in Hamburg. I just want to show you the group in Israel. Some of them, most of them are already gone to other places, but it's my pleasure to show you who contributed to the proto-ribosome project. Ilana founded. Tanaya was able to make the first peptide bond of the proto-ribosome. Gil showed it chemically, and Miri made the first constructs. Miri and Chen made the first constructs that could be made in the lab. The group is run by Dr. Yonath Bashan. She is really the main scientist. And I want to highlight Tamar. that came 18 years ago for 10 weeks. She's still around. And in the day that I took the picture, she had birthday and she baked a cake. You see the cake? Maybe you don't see it well enough. This is her cake. And this shows that for my group, ribosomes are considered sweet. I want to show you my daughter and granddaughter that were very supportive, also my sister, my mother, and so on. My granddaughter gave a speech about me when I got a prize in Paris. As you know, she's a very busy scientist, but she always finds time for me. And she gave me a prize. No, this is the Nobel. Her prize is, for me, at least as important. The grandma of the year is Ada Yonath. And when I asked her which year, she said, which year? You have to prove yourself every year. Otherwise, I take it off the wall. So it's still on the wall. 13 years by now. Ribosomes became popular. This is a carnival. He is me and she. You see, they're dancing. She is a ribosome. Also, young kids became ribosomes. And this is what happened to me. Kishka, who is one of our best artists, drew me. You see, it's symmetrical. The large subunit, two small subunits, two large subunits, two small ones. He's not a scientist. Thank you.
Ada, thank you very much for this wonderful conference. I will put again Fabrício in the room. We have some questions. I want to do the first question, Ada. And there are some proposition. Are there some proposition? There are some proposition about.
Just a second, I hardly hear you. I'll take out the. Yes, okay, now I hear you better.
So, Ada,
Okay.
there are some
Yes.
proposition that try explain the origin of life with other polymers. What do you think of the idea of other polymers before the RNA in the origin of life?
Oh, could be. Could be. I don't know which ones, but could be. I'm not sure that RNA was born together with time zero.
Yeah, but do you think that's so complicated and select one polymer and change it for RNA polymer after this?
No, if there was something, it could be the base for a polymer, not really changing, but developing from one type of bond to another type of bond or another element.
Okay.
This is my opinion, but I don't know anything.
Yes. Fabrício, have a question?
Oh, yes. I have a question about your proto-ribosome ideas and the evolution of the genetic code, because we know there are 64 codons, 20 amino acids, so it should be aggregating amino acid one by one. How do you connect those?
Well, the proto-ribosome could make bonds, probably for RNA. Then the amino acids, they were faster and cleverer and snatched it. And then according to what was needed, I think it was gradual. In the beginning, it was needed to keep the proto-ribosome together, then to move it, then to isolate it, then came the metals. And I think this way the genetic code evolved. Probably in the beginning, it was just 2 letters, or maybe 1 letter for one amino acid. And this took many, many years, but this is just my idea. I have no proof for it.
Well, we have some questions from the audience. I'm going to show one from Carlos Frederico Menke. He is professor at USP. He's geneticist and interested in evolution and origin of life. And he's asking about what would be the RNA needs for the pre-proto-ribosome? Could it be RNA synthesis? Was it necessary?
Could be.
Okay.
Could be, but how it was, I think it was all depending on inorganic reactions.
Okay. So there is another one by one of the students, I think, Isa Mamed, concerning these self-replicating RNA molecules. Would they need a specific environment to replicate, specific temperature, pressure, presence of specific ribosome? You have shown that many environments they could develop, but there are any other particularities?
Yeah, I had one sentence that I carefully put in, that the environment had to be supportive. I don't know what it means exactly, but I don't think that it could be 300 degrees Celsius or minus 20 or things like this, or full of metals or or very, very big storms. But what exactly it was, I don't know. Okay. I can't even say.
There is another question by Professor Gabriel José da Silva. He is talking about a logical issue. Why should RNA build a machine to polymerize amino acids, a function that is already efficiently done by enzymes?
Well, we asked the same question, thank you. And we think that there was an RNA machine for RNA, for either running away from problems or making RNA. But the amino acids are very clever. When they came in, they took it.
I have other question. Do you believe that the genetic code was formed before the appearance of cells? Or we needed the emergence of cell was important to the organization of genetic code, or this organization occurred before the emergence of cell?
I cannot answer you, I don't know. But I think that the genetic code was created by the product together. If there was a good product, like there was this way and it wanted to go in, and this was a good structure, there will be some code for this. Maybe primitive, maybe became more advanced later. Yeah. I'm fascinated by the genetic code. Really, for me, it's something that beyond me. 4 letters control the whole life of the whole world. 4 letters. One of them maybe with a little change. Now, when I say to myself, I think about Shakespeare. Could he write his stories with 4 letters?
Ada, we can say that the origin of the ribosome is the same that origin of life.
The what?
The origin of the ribosome is the same of the origin of life.
I think so. This is my opinion.
Yes. Great.
But we cannot say much. We can only think about these things.
Sávio, there is another question here from Gloria Franco. No, she's one of the organizers, member of the Brazilian Society of Genetics, of the directing board. She's asking about the mitochondrial ribosomes that it seems that Ada didn't mention. They are smaller than the eukaryotic and prokaryotic ones. Do they have the same proto-ribosome site? What are
yes, I mentioned it in the beginning, the same. The active sites, the peptide bond formation and the decoding are the same. The mitochondria are smaller, they have more proteins, less RNA, all types of things, but this is the same. And because there are so many structures now, they can really compare.
Fantastic.
Okay.
Are there any questions, Fabrizio?
Well, everybody is thanking Ada here. Well, yes. Do you have any questions, Sávio?
No. So?
I didn't talk about the 16S.
Ah, wonderful. Can you?
About the 16S? Only what Sávio told me.
right? Sorry, I didn't understand.
About the 16S molecule.
16S.
This is your child, isn't it?
Yeah.
But I think that's completely different origin, right?
Yeah, but it's still ribosomal RNA.
Yeah.
Well, we would like to thank Ada for this wonderful talk, this conference. We've seen that some ideas changed since your last presentations that we attended in these biological evolution meetings in João Pessoa. It was wonderful to meet you and how you are still going on your very interesting work for us, and a lot of people here in the chat of YouTube chat, they are thanking you, for your participation, for your clear answers. It's very nice to have you here. It is an honor for us to have you together us.
And thank you very much for being flexible with the Zoom.
No. Ada, thank you very much. It's nice to see you again.
Soon.
Thank you. We are going to talk Portuguese now.
Bye-bye.
Yeah, you're talking Portuguese. I don't understand this. I even don't understand Spanish.
No, it's Portuguese.
Portuguese. Everybody?
Very similar, yeah.
So thank you for letting me speak English.
Okay. Thank you, Ada. Bye-bye.
Bye-bye. Thank you. You were wonderful.
Thank you.
So, você quer falar, Sávio?
Não, acho que...
Bye-bye. Finito.
Finito.
Acho que foi ótima a conferência da Ada. Eu acho que muda muita coisa no que a gente está acostumado a ver de ribosomo, né? Não sei qual foi a sua impressão, Fábio. Você também já conhecia a palestra da Ada, acho que foi ótimo. Qual foi a sua impressão?
Eu acho que a Ada, né, é um exemplo de dedicação, né, a todo a forma de fazer ciência, né, com todas as dificuldades também, embora ela tenha um Nobel, seja renomada, né, ela era uma pessoa que mantém a curiosidade, né, e eu acho que é isso que a gente tem que manter aceso, né, na nossa carreira o tempo inteiro, né, e ficar atento às novas abordagens, né, às novas metodologias, tecnologias, novas hipóteses, né, e é o que ela está fazendo, né, ela está constantemente, né, evoluindo o modelo dela, né, então é bem interessante essa discussão, que é mais a sua área, né, Sávio? Não é a área que eu participo, né, mas a gente já está escutando, né, e discutindo há muito tempo, né, e a gente vai continuar essa discussão, né, hoje à tarde, com outros temas, né, e até o dia 3, aí, na sexta-feira, contando com a participação de todo mundo aí na audiência.
É isso aí, a gente volta às 2h30, aqui na página do canal. Eu peço ao pessoal da audiência que não entre num link específico, entre direto na página principal da SBG, que lá vai estar o link, porque teve algumas confusões de links. Então, melhor é entrar direto na página da SPG, que lá vai estar qual é a atividade que vai estar online naquele momento. Ok? Então, à tarde, a gente tem a palestra do Fabrício falando sobre evolução humana, evolução e adaptação humana, e logo após a palestra do Fabrício, a gente vai ter uma homenagem ao Francisco Talzano, com um painel sobre a evolução humana com diversos pesquisadores brasileiros. Então, espero todo mundo lá às 2h30. Ok?
Até mais.